Today we're going to be talking about how to analyze cells and tissues with spatial resolution. And the technique for this is called immunohistochemistry or immunofluorescence, depending on how you do the readout. And this is really a nice culmination for how bioanalytical chemists can make progress in understanding biological systems, because it combines a lot of the things that we have talked about before. So as I said, we're trying to get spatially resolved detection in a complicated matrix, a cell and a tissue. And so the way that we're going to do that is by combining microscopy so that we can get that spatial resolution, for example, by using a camera instead of a simple photomultiplier tube to detect. We're going to use affinity reagents so that we have the specificity that we need amongst all the other potential antigens that are in the cell or tissue. And then we can detect using either an enzymatic system with a chromogenic substrate, in which case we call the technique immunohistochemistry, or with a fluorophore, in which case we call the technique immunofluorescence. And I've provided here links throughout the talk, in this case to two sort of overview references, and then elsewhere in the talk, it'll be links to specific examples. So in terms of the history of this technique, it's been around since early in the last century, and it was really established by a scientist named Coons, who was immunizing animals against the targets that they were interested in, for example, against a virus, and then taking the serum from the animal, labeling it with a fluorophore, and then just incubating that whole serum over the cells. And so they would get products like what's shown here, which is a fluorescence from a virus inside of a cell. And so this technique became really popular by the 1960s, and there's a nice historical record that shows many of the images that were obtained in that period, where you could publish a paper basically by going through this process of immunization and then imaging. And then by the 60s, there was also progress in going from directly labeled primary antibodies to what's called indirect labeling, where you use a labeled secondary antibody. We'll get more to that later, but what that enables you to do is have more modularity. And then further modularity and amplification by adding in an avidin biotin linkage into the detection process. And so there was really progress made over many decades, and now we're able to get beautiful images from cells, tissues, and even material science samples with really high spatial resolution. So in terms of what can be imaged with this type of analysis, basically anything that can be targeted by an antibody, including lipids, proteins, small molecules, carbohydrates. And what that means in terms of structures that you can image is you can image subregions within a cell, whether it's the nucleus, the cytoplasm, or the cell membrane. You can look outside of a cell at extracellular matrix elements, and then you can look at tissue level structures, for example, by labeling a protein that's specific to a blood vessel or to a membrane that's between two tissues. This means that there's a really wide variety of applications of this technology. And so what's nice is that it can operate at multiple length scales, either in tissue or within individual cells, just by modulating the magnification that you image the tissue at. And so here I'm showing two images from Genelia Farm, and we see here a tissue level image looking at millimeters of tissue, or cell level images where each of these green outlines is tracing the exterior of a cell, and the blue here is the nuclei. And so in addition to looking at different length scales, we can also look at different types of structures. For example, by labeling actin, you can see the cytoskeleton. And then what's really interesting is that we can get beyond just geography to actually looking at dynamics and function, even within a single snapshot, by labeling for antigens that only appear at certain stages of a process. So for example, by choosing to label for a protein that is only present when a cell is activated in a certain manner, then we can find which cells have been activated. We can also label for proliferation by staining for this Ki67 protein, which is only expressed in the nucleus during proliferation stages. We can stain for phosphorylated epitopes on proteins in order to be able to look at kinase cascades and phosphatase activity. And we can even detect apoptosis and other mechanisms of cell death by staining for enzymes such as caspases that are present within particular modes of cell degradation. So there's both geography and function that you can stain for. All right, so in terms of how these processes work, I've linked here a nice reference from IBD, which walks you through the workflow. And so here what you're seeing is a cell. This is the cell membrane. There's various protein antigens inside it. Here's the nucleus. And so the first step is usually to fix the cell or tissue. Although it is possible to do immunofluorescence without fixation, but it is the norm is to use it. So what that does is it cross-links all of the proteins into place so that they're no longer diffusing. This means that you're going to get higher spatial resolution than if you left things unfixed. And it also means that the process of staining won't change your tissue. If you have tissue, what's not shown here is you would then further segment the tissue into thin slices, typically 5 to 20 microns thick, using a microtome or cryotome, so that you have something thin enough where you don't have to worry about diffusion as a barrier for the antibodies reaching the cells. Based on the goal of what you're trying to analyze, you would either permeabilize the cell membrane with a series of detergents or solvents such as acetone, or you would skip this step in order to leave the membrane intact as it was shown in this image. And you would do that depending on whether you want to stain for an antigen that's inside the cell or on its membrane, like on the exterior. So if you want to look for something intracellular, you have to permeabilize so that those big antibody molecules can get in. We have to block, just like you do in an ELISA, to obstruct non-specific binding of antibodies to to anything that's in the cell besides the target antigen. And then after that, we're finally ready to send in an antibody. So in this example, they've shown a primary antibody where we've sent in something unlabeled and it's binding the target antigen, this yellow protein. And then following that with a fluorescently labeled secondary antibody. And you can see here that we get specific binding to this target. So the secondary, for example, would be like an anti-mouse antibody, and then the primary was mouse anti-human, and then maybe this was a human cell. Okay, sometimes you do something called counter staining, which is where you label other elements such as the nucleus with a nucleus binding dye, and then you finally can do your imaging. So this is a multi-step process. In some ways it's similar to ELISA in that you're sending in a series of reagents, washing in between, and then you finally go to your readout. Just like any other immunoassay, there's decisions to be made up front. For example, whether you're going to try to detect one or multiple analytes within the same tissue, and that will affect what readout you choose and a lot of your methods. And then beyond that, whether you're going to do direct or indirect labeling and fluorescence or enzymatic. And I just want to note that just like in any other antibody-based method, it's really important that your antibody be validated, in particular for whether it can bind to the antigen in the state you're going to be looking at it. So if you're using fixed or frozen tissues or cells, you need an antibody that you know can bind to fixed or denatured epitopes in the sample. Okay, so there's these two modes of readout, direct versus indirect, and here I'm detailing these. Here's the reference from ABCAM. Direct is where you have the primary antibody against the antigen is directly labeled with either a fluorophore or an enzyme. In indirect, the primary antibody goes unlabeled, and then you send in a secondary antibody against it, and that has the label. And so here, for example, this would be, say, that human target, and then you use mouse anti-human primary, and then you would have goat anti-mouse as your secondary. These have various advantages and disadvantages, and I encourage you to make a table right now and jot down what you think the advantages for each of these methods are and their disadvantages in terms of, say, cost, speed, ability to multiplex, and the type of controls that you have to run. So take a moment and think about that, and then come back and we'll go through it. Okay, so let's talk about this. So in terms of speed. Certainly the direct assay, because it has one fewer antibody, is simpler. It has fewer steps. It's going to be easier to automate if you're trying to do that. However, it is less modular in that you have to label every single primary antibody for every single antigen with a fluorophore. And so you either are doing that in-house or you're trying to buy them. If you're buying them, they're at least tenfold more expensive once they're labeled. So it can be significantly more expensive to do it this way. In addition, a direct immunoassay has a weaker signal than indirect because there isn't this amplification effect. So here we only get the number of fluorophores or enzymes that could fit on one antibody, whereas in the indirect case, you can get multiple secondaries binding to a single primary and therefore many more of those readout moieties on there. And so this matters if you're trying to detect a rare antigen, then you would really need that amplification step from the indirect assay. But if you're trying to detect a high abundance antigen or using a very high affinity antibody, then it may not matter so much. Another advantage of the direct assay is that because there's one less antibody involved, there's less nonspecific binding, less cross-reactivity, and therefore fewer controls would be needed here. On the other hand, in exchange for that great modularity and amplification that you get from an indirect assay, you have to do a lot more controls to make sure that you are seeing what you think you see. And you could ask yourself which of these is better for multiplexing. Let's say you wanted to detect 5 antigens at once in the same sample. It would definitely be the direct assay, and that's because it's easier to put 5 different fluorophores each on one antibody than it is to come up with 5 different species of primary antibody and then have those separately labeled secondaries. So you run out of species eventually that you can immunize and get antibodies from. So for multiplexing, it's much easier to use the direct. All right, another decision that we need to make is what type of readout to use, whether to use the chromogenic substrate or a fluorogenic or a fluorescent label. Here I'm showing you the product of an enzymatic substrate or an enzymatic label with a chromogenic substrate, and you can see here we've got 2 colors, one that's brown and one that's sort of this bluish purple, and these come from 2 different substrates that you can use with the enzymes that precipitate out in different colors. It's actually hard to get too many more colors than this, and so enzymatic immunohistochemistry is typically limited to only 1 or 2 analytes per sample, and then you read this out with a color camera. The resolution of this is somewhat limited if you're interested in subcellular structures because the substrates can diffuse a little bit before they precipitate out onto the sample where you need them to be. And so this wouldn't be good, for example, for a single molecule analysis, but it works great for cellular level or tissue level analysis. And a real advantage of immunohistochemistry like this is that the precipitates that are formed are very stable over time and can last for years. And so these types of analyzed cells and tissues can be banked and then returned to again and again many years later. And so people like them for that reason. On the other hand, immunofluorescence can have advantages if you're interested in very high resolution, even single molecule imaging, because the fluorophore is directly attached to the antibody. But its main advantage, and the reason that people often choose it, is for multiplexing. So if you're trying to look at more than 1 or 2 analytes per sample, fluorescence is really the way to go, because you're only limited by your detection system with how many fluorophores you can detect. Unfortunately, though, because of photo bleaching and room light, the samples are often not stable for more than even a few days, and so it's important to image them right away after analysis. Okay, so there's a lot of controls that go into being a good analyst with immunofluorescence or immunohistochemistry. Otherwise, you may be getting signal, but it's not telling you really where the analyte is. And so there's a series of controls that you would do. You would almost always do what's called an unstained control, which tells you whether there's bright autofluorescence in certain parts of the tissue that needs to be subtracted out or otherwise accounted for. There's types of controls where you leave out each antibody one at a time. For example, what I'm showing here is a indirect assay where they had a primary antibody labeled, shown here with green fluorescence, but the green fluorescence is really the product of a secondary antibody stain. And so if you leave out the primary antibody, which is what's shown on the right, you don't get any of that green fluorescence, but with the primary antibody, you get it. And so this example is one where the signal is very clean. There is no green signal visible in the control that lacks the primary antibody. So that's a good outcome. On the other hand, if there was really bright green signal here, we might say it's because of autofluorescence or because of nonspecific absorption of the secondary antibody. There's other types of controls that you can do, such as isotype controls, which we mentioned before during flow cytometry, where you're checking that say the rat IgG1 that you're using doesn't bind nonspecifically to FC receptors in your sample. This is more important in some samples than others if they have a high abundance of FC receptors. And then you can also do controls where you either lack the analyte, so a negative control, or you've obstructed the analyte by adding something in that will block it initially. And then the flip side of that is a positive control, a sample where you know that the analyte is present and where it should be. And that way, by comparing the negative and positive controls, you can really be certain that you're seeing what you want to. Okay, so it's worth taking a moment to think, when would I choose microscopy or flow cytometry to analyze, since in some ways they're similar. They're both using antibodies and they're both often using fluorescent readouts. The big advantage of microscopy is for when you need spatial resolution. So for example, if you want to be able to watch cells or molecules motion over time, or expansion of cells over time, then you really need a spatially resolved readout that you can get with the camera or with a scanning PMT. On the other hand, you would choose flow cytometry for certain if what you needed was not spatial information, but rather population level statistics on the expression of a variety of markers. And so flow cytometry really excels at very high throughput analysis with thousands or tens of thousands of cells per sample, where you get a large number of parameters for every single cell. In contrast, microscopy is often quite a bit lower throughput. If you were trying to do microscopy-based single-cell analysis, it's a challenge to get more than a couple hundred cells at a time, especially if they're being imaged manually, versus the multiple thousands that you can get with flow cytometry within minutes. Okay, so these are really complementary techniques depending on what types of questions you're asking. All right, so finally, I invite you, if you're interested, there's a variety of resources on the internet where you can look at different types of images that have been taken using these techniques, including some from the NIH and others from various vendors. And so it's worth taking a look at, sort of to quiz yourself, is to pull up one of these images and then read the methods that are associated with it, and then see if you could sketch out that immunoassay that was done in order to make it work. But it's also really fun just to be able to see all the types of structures and analysis, because it can give you ideas for your own experiments. Okay, that's it. So I'll see you in class later, and I hope this was a useful introduction to immunofluorescence and immunohistochemistry.